Myers. I'm our LIFT project administrator. I'm also the administrator for STEAM Pathways. And I'm really excited. This is like I think, the coolest event ever because this showcases a little bit of what our SPEAR team and others who have participated in any type of research and projects. But I have the great opportunity to introduce to you our awesome coordinator for STEAM Pathways, Cassie Coiner. She's got a lot of great things to share with you all, and just want to let you all know of our SPEAR team and other students that were involved in all of this stuff that I'm really proud of you for all the work that you guys do. Thank you.
All right, good evening, everybody. Like Crystal said, my name is Cassie Coiner. I just wanted to do a quick welcome, and then we're going to get started with our awesome students that have some really great research projects to share with you guys. So a couple of the things we have coming up, we're kind of getting ready to go on a break because it's the end of our spring term. So this is kind of one of our big wrap-up events. Coming up in August, we have our STEAM Academy, which is going to be August 10th through 13th. It's going to be from 9 a.m. to 4 p.m. It's more of like a day camp. So it's for 6th grade through 12th grade. And we have some awesome activities planned this year that we haven't done in the past. And we have some flyers for that up. Registration for that will start on June 15th. And then another really cool thing we're excited about that we're actually fundraising for is called our Student Spaceflight Experimental Program, where we're going to hopefully recruit students that are juniors and seniors and college students in teams of 2. And we're hoping to have at least 10 teams so that they can create experiments that will actually get the chance to go. One of these groups will have an experiment that will actually go up on the International Space Station in the spring. So that's going to be Mission 22 coming up. Hopefully the launch will be in spring of 2027. That could get pushed back because, well, we know how weather in Florida likes to work sometimes. So we are fundraising for that, and there are some flyers for that up there as well that has our QR code if you would like to donate to help us raise our funds for that. We are on our way to our goal, but we could still really use some help, and then we'll keep you guys updated as our teams form and we get the process start for that. But without any further ado, I will let our first presenter, Sabrina, come up to share her research.
Hi, my name is Sabrina Pingree, and I'm an OSGC student ambassador at Southwestern Oregon Community College. Tonight, I'll be talking about expanding STEM and space science access in our rural community through outreach, advocacy, and hands-on learning experiences. The goal of this work was to increase STEM accessibility and engagement in communities through inclusive, hands-on outreach experiences. I organized 13 outreach events and reached 586 participants ranging from pre-K through high school, college, and community members. The activities included polymer chemistry, making slime, a pH and ion experiment with red cabbage at the fire hall, STEM story times, an interactive physics lesson, and astronomy outreach. The locations where I did these events were at schools, the local library, and community events hosted by SWOCC STEAM Pathways. And also I held events right here on the SWOCC campus. I also conducted astronomy outreach using a portable planetarium, courtesy of SWOCC STEAM Pathways, and also a sun spotter solar telescope courtesy of the physics professor, Dr. Coiner, and then a C-STAR digital telescope loaned to me by Oregon NASA Space Grant Consortium. And for these events, a lot of the students that went to them, it was their first experience to astronomy and space science, specifically at the schools. This telescope picture over here is of the Orion Nebula that was taken on the C-STAR digital telescope here at SWOCC during the SWOCC STEAM Pathways Artemis II launch party. One important experience was working with the school that has mostly neurodivergent students, as the principal told me. And that really reinforced to me how in rural areas it's important to be inclusive and have hands-on STEM environments that promote accessibility and representation. Here you could see I'm at a SWOCC STEAM Pathways event, STEAMoween, helping a parent and some students learn about using ions with balloons. And then in the other picture, I'm at the library reading a STEM storybook to kids. This work also extended beyond outreach events through an OSGC presentation here on campus to my communications class. And then I was also given the opportunity to go to Washington, D.C. to go to the National Space Grant meeting, where I was able to talk with other students and affiliate representatives from across all 50 states about what they've been doing through the National Space Grant Program. And I also had a unique opportunity to be introduced to the associate administrator and the deputy associate administrator of NASA's OSTEM. I was also able to go to congressional offices on the Hill and talk with members, Oregon members, about the work that OSGC does, why they do it, and how it's important. Something really amazing that I was able to do in partnership with SWOCC STEAM Pathways was organize the development of the only countywide science and engineering fair. And SWOCC STEAM Pathways plans to make it an annual thing now. And these are two of the students that were at the science and engineering fair with their participation certificates. Unfortunately, I wasn't able to get donors to donate things for them, so I made those, and then SWOCC STEAM Pathways administrator went and bought them some prizes, which was they thought were really cool. Yeah, and they loved it. So on the left, we had a project about can tardigrades really survive in space by putting tardigrades in a freezer and then looking at them through a microscope. And then on the right, we have a student who did a physics experiment about fluidized bed. And then we had one other student that participated as well. We had a very small turnout this year just because it was the first one, but I'm expecting there to be more students to come next year once more and more people hear about it. So overall, I had 13 outreach events with 586 participants. And here's a quote from one of the students at one of my events, which I thought was pretty cool because I also grew up in a rural area with not a lot of opportunities. So just hearing that from them really touched my heart. I really liked it. So overall, the key takeaways from my project were access matters and that representation matters just as much. And another thing that I took away is that hands-on learning increases engagement. So just talking at students isn't as effective as actually doing hands-on activities. And that STEM outreach supports future STEM pathways. Just want to say accessible STEM outreach doesn't just teach science, it helps students imagine themselves belonging in science. And I want to give acknowledgements to Oregon NASA Space Grant Consortium for funding and also support, to Southwestern Oregon Community College faculty and mentors, especially OSGC affiliate representative Dr. Aaron Coiner, for helping me out with events and giving me advice, and also STEAM Pathways Administrator Crystal Hopper Myers for telling me to apply for this program. And I also want to give a special thanks to Cassie Coiner, because I could not have done a lot of the events, especially the science fair, without Cassie Coiner. She helped me out a lot. And she's a STEAM Pathways coordinator. I also want to thank community partners for their collaboration, as well as the students and the families who participated, because if they wouldn't have participated, then the events would have meant nothing. So thank you, guys. Alongside STEM outreach and advocacy, I've also been involved in chemical research at SWOCC. So for my next presentation, I'll be discussing the work done on lipid extraction from fossil-associated sediment. Okay. So this presentation focuses on our research project titled Breaking Bonds, Lipid Extraction from Fossils and Surrounding Rocks. The project began through the SWOCC STEAM Pathways SPEAR program. Samantha Hayes and I pursued the opportunity and began theoretical research and poster development with funding from OSGC back in the fall of 2024. The goal of this project was to extract lipids from fossil-associated sediment and characterize the fatty acids present. So why did we choose lipids? Well, lipids preserve well because of their long hydrocarbon chains. So even after millions of years, when DNA and proteins degrade, lipids survive. And then they provide us information about biological and environmental conditions. So this is a picture of the lipids that we extracted from fossils in water, so it's easier to see. And this is the fossil that we use for our research. So this is a whale hip bone that I believe Dr. McLaughlin said was about 10 million years old. So as you can see around the hip bone, there is a dark yellow around it. And many fossils show a dark biological halo like that in the surrounding sediment, which scientists think may form from lipid migration into the nearby rock material. So our overall workflow for doing this, we started with SWOCC slit extraction, and then we moved to saponification, and then thin layer chromatography, or TLC. Column chromatography was next, and then our analyticals equipment, NMR, and FTIR. So I'll just explain what all those are. So first we started with SWOCC slit extraction. SWOCC slit extraction is the one on the left. So it's a continuous solvent extraction method where we use chloroform and methanol to isolate total lipids from sediment. And then after that, we put it in the equipment to the right, which is a rotary evaporator, to get rid of all the hazardous chemicals from the solvent that we used originally. And after that, we were left with this one on the top. So we move on to saponification, which breaks triglycerides into free fatty acids. And we use ethanol and sodium hydroxide under reflux conditions. And then we use hydrochloric acid to free the fatty acids. And then the rotary evaporator again to get rid of that solvent. And you guys might have heard about saponification before. It's the same process that people use to make soap. So we did make some soap in this process. Next, we had an analysis technique called thin layer chromatography. TLC helped us test the polarity differences in the lipid mixture using multiple solvent systems. The results showed chemically diverse lipid fractions, which we got through putting iodine in a beaker and then heating it up. So that's what that beaker is and that purple gas. It's toxic iodine gas. So we had to cover it up. And as you can see, the top TLC plate had about 6 different circles. And then the one on the bottom left had a lot more. And the difference is the different polarity ratios. So that's just showing that there's a whole bunch of different kinds of lipids in our sample. Next and currently, we're using column chromatography, which is separating liquid fractions using silica gel, which is that white stuff in the middle of the column. And this is also called adsorption chromatography. So we used a solvent polarity gradient. So we started with hexane, and then we started with 100% hexane, and then we did 50% hexane, 50% acetone, and then we moved on to full acetone, and then acetone methanol, and then 100% methanol. This is just to separate all the different types of lipids that we found out from TLC. So currently we are doing this in the lab, and we're letting the fractions just evaporate on their own because the solvents that we're using evaporate pretty quickly. So far, we've successfully developed a multi-step extraction and purification workflow. And we've also collected separated lipid fractions for future characterization. Our next steps are using the NMR and FTIR machine. So we're going to be using nuclear magnetic resonance and Fourier transform infrared spectroscopy to confirm the different fatty acid structures that we have derived from the lipids that we have derived from the fossils. So just to recap, the project began with Samantha Hayes and myself through theoretical research and early lab development. And then I continued the experimental workflow and method refinement with the help of our high school intern over the summer. And then more recently, we had additional contributors assist during later lab phases. So in conclusion, fossil-associated sediment contains extractable lipid signatures, and our workflow successfully isolates lipid fractions for future analysis. And also our OSGC Orion interns are currently continuing the work. So that's Amy Gonzalez and Mary Gold. I'd like to thank Wynn McLaughlin, Derek Morelli, the Chemistry High School intern, the Chemistry Club, OSGC Orion interns, SPEAR, and the Oregon NASA Space Grant Consortium for their support. Thank you. Our next presenter is Amy Gonzalez. If you guys could give her a round of applause.
Hi, good evening. My name is Amy Gonzalez. I'm a freshman here at Southwestern. I was given the opportunity to continue this research project that I started back in December to present for you here today. So it's called Filling the Space in Cancer Research. So this was just something that I started in December, and something that intrigued me about cancer research in space and the certain, I guess, systems that they do research. So it is kind of known that cancer is the second most common cause of death in the US. We are working towards more innovations and treatments that are helping reduce the impact and then preventing the disease. So exactly how is space involved in the treatment of cancer? The two main concepts that I will be looking in tonight are 3D tissue chips and protein crystallization. So the picture up on the corner is an actual 3D tissue tip. They are quite small and they're made of a flexible plastic. And then those two pictures there, which I will be talking about a little bit later as well, are what protein crystallization looks like. So as you can see in microgravity or in space, they're a lot bigger compared to Earth's gravity. So 3D tissue chips are small microphysical systems. They replicate certain organs. You can choose different organs. And then they test and they research on how they respond to environmental factors as well as systems in the bodies and maybe even other sorts of drugs. So in December of 2018, NASA did launch a mission with these 3D tissue chips. So they mainly were focusing on how spaceflight affects astronauts. So they're looking into muscle wasting, immunosuppression, cardiovascular deconditioning, so using up the heart, decreased bone density, and even small amounts of radiation. This picture shows that 3D tissue chip again compared to a quarter. So like I said, quite small. What they look like with processing, they do run different tissue chips for different parts or different organs. So liver, kidney, lungs, and they each run in different processes, and that's how they analyze the different parts. And then it is quite large to run it, as you can tell. So with the lab and running all of these different systems, it starts about about 1350, and then for launch they need to decrease that immensely, and then even in orbit it has to be even smaller, which will be talked about later as well. Along with that is protein crystallization. So that is taking proteins within the body for disease or medicine and crystallizing them to gain a 3D structure, which we can further analyze. So, like I said, with microgravity, you take away the suppressant that is gravity. This allows proteins to grow in a much higher quality structure. And with that, we're able to analyze them a bit further and actually look into what we can do to create medicines as well as analyze these for other diseases as well. So specifically, there was a research by Microquinn and Biotech where they were given the opportunity studying breast and prostate cancer. They sent their project, sorry, they sent their project to space, and that's where they specifically crystallized the TMB-IM6 protein. A little bit complicated, but they were able to analyze that, and specifically, they were able to highlight that certain pathways are more important to cancer development than others. They looked into things such as intracellular environment, or ICE. It's a structured space within the cell membrane, so the outside of the cell, and it is also responsible for the cell's life cycle, so creating and killing the cell. So with that, microgravity also sparks an increase in the productions of genes that create these regulators. So they're hoping to create more medicines and finding ways to create structures that bind to them in order to control them and eventually hopefully kill these cancer cells. This photo shows a very popular astronaut as of recently, Christina Koch, who was working on this microquinn project. So these photos are also part of that biotech project. It shows the process in 24 hours of the medicine that they were creating. It specifically targets in the endoplasmic reticulum of the cell. So as you can see here, that's the endoplasmic reticulum, and slowly more holes start appearing in that, which means it is killing off the cell. And towards the end, you see the dead cells, the dead cancer cells. So slowly and slowly, it kills it off. Another very important part of cancer is the CRAS gene, which drives 30 to 40% of all cancers. It has a specific protein it's a signal pathway for most cells. It either tells it to create and just become a normal cell that runs through the body, or take on a specific job and mature, have a specialized function. When mutated, this class of gene, which is oncogenes, it has the ability to turn normal cells cancerous. So this picture right here is actually a crystallized CRAS gene. The two red circles are the two most popular areas where these mutations can actually occur. Again, here's that photo from the beginning. It is the crystallization of the CRAS gene, and again, just emphasizing how much clearer and larger they look in space compared to Earth's gravity. And so when these turn cancerous, it can lead to immune evasion, and tumor growth. Another very important part are endothelial cells. So these are actually the cells that provide blood to the tumors to grow. Here in the space, we have Serena Nyon, who's was working with a project where they sent these cells to space in order to try to find ways either kill them off or deplete them of oxygen and nutrients so that they're not able to help these cells or these tumors grow. So this is a picture of those cells. Very pretty. So some of the therapeutic medications that we have found involving these genes and proteins. So specifically with the CRAS gene, they have been working with immune checkpoint inhibitors, ICIs. So basically, they're working more towards the environment around cancer cells rather than the cancer directly. That way it can prevent the spread before directly killing the cancer. Continuing on, they have the THP1 cell line, which is not exactly the cancer, but again, working with inflammatory and responses. And in that way, they're trying to protect the area around the tumors or work with that area first. And then specifically, again, with the TMB-IM6 protein, again, like I said, it was located in the endoplasmic reticulum. Doing RNA interference, so controlling what the cell does. It can suppress its expression and even reduce the tumor growth. So creating this, again, will help with the environment around those tumors and cellular injury. And this is just a photo showing that process of the RNA interference. So here's the protein, and then sending the other sorts of protein to change what the cell does. So some slight limitations to the research. Like I said, the space aboard, I'm sorry, the ISS, the cost to run the experiments, and then most research is most based in vitro compared to vivo. So conducting experiments outside of the human body, not involving humans just yet. So with the space, like I showed in the photos earlier, they are working towards reducing how big these labs are, and or finding ways to just singular take out specific pieces that they want to look into first, maybe working towards sending more up later. In terms of cost, so the main, I guess, mission with the 3D tissue chips was to diminish the expenses that happen with failed clinical trials. They are very, very expensive, and you don't get much answers. So with that, the 3D tissue chips, although expensive and protein crystallization, as well as sending it to space, it is an investment into helping close this gap between vitro and vivo, basically making better medicines, seeing how they're able to respond to the body rather than putting them right into the body. That way, diminishing that clinical trial fails, and yeah, just creating better medicines and more targeted medicines. Inevitably to save in the future. And then this is just a small photo, each chip for a different part of the body. So all of this also helps with research beyond cancer. They're doing treatments for certain liver and lung diseases, helping with muscular dystrophy, and even looking into the chemistry of psychiatric medications to even look into mental disorders. So overall, I did have to base this research mostly off of NASA's mission, so driving and enhancing knowledge, innovation, economic vitality, and stewardship of Earth. There are no solid direct answers with this kind of research, no like heavy medicine that's helped cure cancer or anything like that, but it is all steps towards finding more answers and finding better medications to inevitably help. And that is all. I want to say thank you to, thank you to SWOCC and to my professor, Dr. Coiner, for giving me the opportunity tonight. They can make tissue chips that are like specified to an astronaut, like considering their different, like if they have certain things that are wrong with them to begin with. But for the most part, they just take on the function of an organ. Mhm. Thank you. Anything else? Mhm. Um, I think the most interesting to me, it would definitely have to be the specific proteins. I know that's kind of broad, but like the TMBIM6, it just has so many functions, and to be able to take something so small and look into it to even change how a singular cell can function, I think has been the most interesting part to me about all of this. Thank you.
Hi, everybody. So my name is William Snively, and I was an intern last summer under Dr. Coiner for the STEAM Pathways internship with 3 other interns. And so our goal was to build a resistance training tower for the SWOCC swim team, and the goal was to make one that was cheaper and just more efficient than the one that they have. And so we wanted to make it more compact, cost effective, and build a working prototype at the end. So the process for doing this started with the research. So we had to look at all the different designs they have out there, how it works, the materials, the different specifications, and then the structural integrity. So we had to run some tests and make sure that we could actually build something that would work fine for what they need to do. And so the next step, which was probably the main thing that we did and took the most time, was designing it. So we brainstormed with each other. We each came up with our own ideas, and then we would sketch out designs and use CAD for 3D design. So that is my 3D design that I came up with. And I think the one that we ended up kind of going with for our prototype and to try to make. And so after that, we did collaboration. So all of us interns, we shared our findings, and then we gave feedback to each other to figure out what we could do differently and do better to make the best design. And then we did this with weekly meetings and discussing over Discord and messaging. So the next step after that was to prototype. And so we had to order materials and then get a price estimate and then prep the building. And then Graham, one of the other interns, was going to 3D print pulleys. And so the next steps would to be a bill of materials, which the main materials are going to be aluminum tubing, the polypropylene rope, a 20-gallon bucket or some other thing to hold the water, pulleys, and hardware. And so in the end, we could make the training tower, which would work by basically putting a belt around the swimmer, and then you fill the bucket with however much water you need to get a specific weight, and then they can swim out. I believe it's 50 meters and then back, and that'll give them enough resistance to get stronger in their swimming. And so the cost of this would be roughly $1,400. The aluminum would be the most expensive part, and then the rope, the bucket, the pulleys, hardware, and all the other miscellaneous items, they're just smaller costs. And so the build plan, we have to create blueprints, a list of instructions, and then we have to submit those to the SWOCC welding department, which we haven't done yet because sadly last summer we ran out of time. So maybe that'll happen in the future. And what I've learned, so last summer throughout the 8 weeks doing it, I was able to better my skills with 3D design. I worked with the other interns and got better at teamwork. I learned to observe and research stuff, and I gained a lot of experience in engineering, which showed me that I do want to do that in my future and hopefully get a career one day with engineering. And I would like to thank SWOCC and Dr. Coiner for making all this possible. Thank you. Yeah. I used a little bit, but that was probably the most I've ever used it before. So it was good to get practice with it and learn how to use it. Yeah.
What engineering degree are you pursuing?
I want to go for mechanical engineering with like an aerospace focus. So hopefully I'll go here to SWOCC and then transfer to OSU or something. Yeah. Yeah. Aluminum is the most lightweight and it's corrosive resistant, so it wouldn't corrode with the water there in the pool. And it's fairly cheap. I mean, it's not the cheapest thing, but it's probably the best material that we could come up with. Yeah.
Kind of simulation, like analysis.
I believe Graham, one of the other interns, tried to do that. I didn't do it because I don't have a software for that, but we I think did try to do that, and if we didn't, we will try to do that in the future if we have the chance. So yeah. Yeah.
What was the, during the
I would say the biggest challenge was probably time because we didn't end up having enough time to actually fully build the prototype and like fully achieve the goal. Like we still got our design done, but we just didn't get to finish it completely. So yeah. Yeah, I would do it, but I also need the other ones to yeah.
Costs compare if you were to purchase this apparatus?
Oh yeah. So I believe the one that the swim team currently has is around 3,000. So they're typically pretty expensive, and so this would basically cut it in half if we built it ourselves.
model, like 3,000, 4,500.
Yeah.
Part of that, two bucket system, so they only do 2 lanes at a time, whereas the cheaper method, you're gonna be able to, we were gonna be able to actually build them specifically for the number of lanes that we have, so you could essentially have more swimmers.
Mhm. Yeah. Okay. Thank you.
The next, talking about their prototype and Northland.
Okay, so I'm Emmanuel Salinas.
Joe Molina.
Yeah, and so our team name for the Invent Oregon is research project that we're doing is a temp reflex. So here we go. So this is a little bit of context of the research and more opportunity that we've been given. So first, our slides are gonna have context, our timeline. We're gonna talk about customer outreach because that was a big part of our project. Our prototype and our current progress, and then what we're doing going forward. So I'm gonna introduce it with this quote: 30 million users expected by 2030 in the United States alone. What does that mean? Well, this comes from an article of JP Morgan that talks about the GLP-1 medication market and how it's growing and increasing year by year. And that is only specifically talking about GLP-1. Home administered medication market is increasing each year along with insulin and other medications. Now, with that, there are $1 billion in cold chain packaging because there's been a shift from retail to delivery. It used to be that people used to have to drive to the pharmacy or go to their doctor's office to pick up their medication, but now it's being delivered at home. Next up, I want to say that up to $1,300 lost because of one simple mistake. Now it's getting more into focus of what we're actually doing. And that's trying to avoid improper storage. Say you leave out a milk carton and it goes bad. That's okay. It's just a milk carton. But if that was your medication and you needed it, more than that, it might cost you a lot of money, like this standard cost for a GLP-1 medication, but it can also harm you and be unsafe to the user. And so we saw that this as a problem, and we developed a roadmap to try and solve it. So first, we needed to find our problem. You know, we needed to identify our customer and really fit their needs. Then we began designing our prototype, and that's what we're on right now. And we're trying to make a final product, and we're creating to finally make a working product that benefits the consumer. And now I'm gonna pass it on to Jomar to talk about customer outreach.
So a big thing that Invent Oregon taught us when we went to boot camp up in Portland was what your customers want and how your product is gonna adapt to their needs. So the three things that we're looking at are identifying the worries of our customer, their common habits, and what they want directly for our product to do for them. So recently, we sent out a survey to uh, the public, and we got some results back. And I'll just go through the questions. What was the main reason your at-home medication needed replacement? And majority said they were unsure if that medication was still safe to use. Second question being how, if you ever worried about refrigerated medication being left out for too long or stored incorrectly, and majority said yes. And lastly, this question is specifically to what we should do for our product, being which reminder or alert method would be most useful for medication storage. And majority said phone notification and email and text message. So our solution, hopefully Temp Reflex is a temperature sensitive alerting system that reminds users to ensure proper storage for their medication, at home administered medication. And with that, we want to, for the product to work, we would attach our device to the packaging of the medication, which would read the ambient temperature. And if there's any deviation from that temperature, the user would be alerted through a light, a sound, and an app notification. And for our product, we really wanted to ensure like accessibility for all users. So that's why we have 3 different alerting systems. And in the future, we want to include a tracking feature for out of a safe zone for the temperature, which Emmanuel will talk about.
Yeah. So what we learned when we went to this boot camp, because Invent Oregon isn't a competition, that they put a big emphasis on really understanding what the customer wants and not what we want or what we think the problem is. And so we found from our surveys that a lot of people wanted this phone notification. And so we thought, okay, let's make it a sort of app along with the analog. And what the safe zone is it tracks the amount of time that the medication is spent at a unsafe temperature. Because usually these medications are okay to be left out for a couple hours and sometimes even a couple days. But over time, this can reduce the efficacy of the actual medication and create unsafe use to the users. And so we think that tracking that time allows the users to not be unsure whether their medication is safe or not and eliminate that problem. Yeah. And so now we're going to talk about our prototype.
So the picture to the left is a very rough sketch of what we want our product to look like. It'll be pretty small and attachable to the packaging of the medication. And we want to use, we want to have the housing to be 3D printed, but also ensure that it is moisture resistant and stuff, things like that. So it's able to be in refrigerated temperature and have, and let it be safe to use in moisture environments.
Then on the left, we have the circuit sketch that we made. This is a simple analog design of the temperature sensitive light system. And so this doesn't really play in with the sound or the app, but we can draw similar conclusions from the circuit. And so the thing I'm going to focus on the circuit is there's an LED, there's a thermistor, and there's a potentiometer. And this potentiometer is adjustable. And so essentially, if we adjust it to a specific position, say you put this circuit inside of a refrigerator, and we adjust it so that when it's at that temperature, the light won't turn on. Once it's taken out of the refrigerator, the thermistor senses a change in temperature and allows current to flow, making the LED turn on and therefore alerting the consumer that they need to store the medication back to the proper temperature. Yep. And now,
So this picture right here is a picture of one of our pretty early prototypes of our product. In the picture, we are using a Arduino circuit and LEDs, resistors, and a temperature sensor. And that little black thing on the left is the temperature sensor. And basically how this circuit works is that once that sensor detects heat, the resistance will go down, causing the light to emit. And something that won't probably won't make it to the end product, but I thought it was pretty cool was the lighting system. So the first one would turn on, it'd be normal. Second one would be a warning, and then the third LED would be your critical temperature. And yeah. And there's also a lot of coding that goes behind this, but we'll just skip on that.
so going forward, how to turn this prototype into something that a consumer can actually use. We want to create an embedded control system. And so that's part of what we're using with our Arduino, because the analog circuit that I showed earlier, um, yeah, it works, but we need to make sure that it's all controlled by itself and that the user has the most accessible experience as possible. And with that, we want to print a minimalist, easy to use housing. So making it easier for the consumer to use. And so we're going to 3D print that. And then towards the end, because this is a competition, we want to make sure to pinpoint our target consumer, because they were big on, Invent Oregon was big on specifying who's most likely to actually buy it, not who want to buy it. And so we need to adjust our prototype to their needs. So this is our current timeline.
So currently, right now, we're in our prototyping and customer outlook phase. Right now, we're reaching out to clinics, hospitals, and people of the public to see what we should do for our product and what they think would be very helpful. And then early June, we're gonna have hopefully a finished product and have things set up before finals. And then on the 25th and 26th, we will be competing at the finals, which is in Bend this year.
Yep. And so if you want any more information, these are our emails. If you have any questions, feel free to ask. But this QR code here on the left is an anonymous survey. And if it relates to you and you'd like to fill it out, we appreciate your feedback. We're trying to get more customer discovery. And yeah, thank you. Yes. Uh,
Then, or you send it back, like, how does it work?
And so we forgot to mention, so this is a USB C type charging, so it's rechargeable, and so we're not sure how long it will last because we still have to consider the limitations. We want it to be small, and so that might impact the amount of battery life we can store in it, but it will be rechargeable in the end. Yeah. Yeah. Yeah.
Great, great sailing.
Yeah, so really turning it, we're gonna eventually have to turn it to like Bluetooth. And so making a circuit is a little bit easier um, when you don't have to connect it to your phone. And so we're trying to get a hold of that, how to use the Arduino, because it's our first time using one. And so we also gotta, the one that we saw in the picture, that one's huge. We gotta turn it into tiny ones. So there's a mini one, and we don't even know how that works. So yeah. Oh, Mark.
Ask about Bluetooth. It does make a Bluetooth module. I don't know if you've experimented with that or not. Using, yeah, Bluetooth signal, to connect your phone.
Yeah, absolutely. Yeah. Since the first start of prototyping, people that we talked to about the product, they were like, "Oh, can I connect to my phone?" And so, yeah, it's a pretty big deal. Yeah. Anyone else? Yeah, Miss? You're good. You're good. Mm, yeah. Yeah. No, that you bring up an interesting point because a lot of sometimes these medications are given to people who don't have access to like a phone or don't necessarily know how to use it. And so having a caretaker not be in the room, yeah, that's a good point. Yeah.
Um, the different ranges of temperatures.
So yeah, that comes along with the Arduino because with the potentiometer, you have to manually adjust it. But with the Arduino, you just kind of have to tell the computer. Um, and I guess we have to make an integrated app system where you can adjust it directly on the app. Yeah. Yes.
Value added packaging for the big pharma.
So that's something that we're looking to discover with our customer discovery because at first it might seem okay, we want the user to buy a product, but then is that really realistic? Is the question we have to answer because um, a lot of the times customers will be like, well, I don't leave my medication out, so I'm not gonna buy it. And so they leave that risk in the air and they don't buy the product. And so with focusing on big pharma, we could tackle insurance claims. Oh, they can't ask for a replacement and cost us more money. And so yeah, right now we have to figure out who's our main target. Big pharma is one of the people we're considering. Yes, Mark?
The calibration on your phone is insert.
Mhm.
You know that it is at, that it's at when it's reading, when it's at.
Okay.
Arduino code that you're getting.
So at first, I thought that the thermistor that we can use like that we had in lab would would work just fine to measure temperature. But then when it gets to specific temperatures and when it comes to medications, we have to be very like um, aware that they have to be right and they have to be exact. And so Arduino makes this makes a lot of things compatible. And so we bought we just came in the mail today actually, is a temperature sensor that is highly accurate and connects to the Arduino and that good code. Yeah. Any more questions? Yes. Mhm.
But are you, have there been reality, like if you're advising consumers to take these medications, saying that they're safe, is there something? And does that make sense?
Oh, could you explain a little more?
Do you have any concerns about like, are you put legal disclaimers on the app saying like,
Mm.
or
Like we're not responsible for it. Oh, okay. I mean, I'm not sure.
I guess we haven't really thought about that yet because we're not too sure if we want to actually bring this to the market, but it's definitely a good thing to look into. So thank you for that.
Yeah. Okay. Well, thank you.
Hi, I'm Mason Miller. I got the opportunity this term to work on what's called a STAR program. It's Student Academic Research Review. It's done by OSGC. And what we do is it's a paid opportunity where we get to investigate a certain aspect of OSGC's goals, like the current mission statements that they're trying to achieve, and we get to look into something around that. And I wanted to look into manufacturing in space, and specifically 3D printing in space. So there is a massive problem with space flight, and that is if you want to create new tools, say a need arises, like, oh, I need this wrench. We didn't pack this wrench. The wrench broke, right? Or if you need to make some sort of part to replace something, oh, a gasket breaks, a fitting, something like that. It's impossible to predict what's going to break because you're kind of expecting it not to break, right? And if you're in extreme isolation, like if you're on the International Space Station, you can't just like, hey, send one up, because you have to load it onto a rocket, and that has to be sent up, and that has to dock, and you have to deliver this part, and it takes time. And if you're having an urgent emergency, that wait time could mean life or death. And most processes for making these parts aren't possible in 0G. If you think about casting metal, right, that doesn't work in 0 gravity. If you try and pour it, it's just going to fly everywhere. And that's where 3D printing allows for a way to navigate around this. You can pack filament onto a ship and then ship it to the station, and that filament can be fashioned into any sort of tool or part you need with some tangents. The process is relatively risk-free, and because it doesn't use high pressure, it doesn't use high speed, it doesn't use really high temperatures. It's safe to use in a very dangerous and hazardous environment like a space station, and it's very efficient. It minimizes a lot of waste. So like I was saying, the way it's going to work is you would take the filament, which is usually long coils of some sort of material. We'll get into the different materials that you can use later. And you're going to put that onto a rocket and ship it up. It's really, really expensive to ship anything to space. So packing things as densely as possible is very important, because every pound and every square foot, every cubic foot matters a lot. There are a few downsides to 3D printing. It's not an end all. 3D prints are weak along the layer plane. The way a 3D printer works is it deposits a layer of material one at a time until your entire material or your entire object is built all at once. And what that means is each layer plane is weak across that way. If you were to 3D print a bar, making it standing up with each layer like this, it'd be really easy to snap the bar because that's the direction of its layer planes. It comes with some unique challenges. It can be difficult to get repeatability on some printers, especially with plastic, as we'll see. And not just because of the layer lines, but indeed just because of the manufacturing. They are almost weaker than traditional manufacturing, like if you're going to mill it out or cast it or cut it out of a certain material. And the first most common form of 3D printing is FDM printing, which stands for fused deposition modeling, and it's most commonly used for plastic. So we use a kind of plastic called thermoplastic that gets softer the more you heat it, and it gets stickier the more you heat it, so it adheres to itself when it's hot. And like I was saying, each layer that you place down will build upon the last in a linear fashion. This is the first 3D printer that was placed aboard the ISS. It was the first one to be tested in 0 gravity. I believe it was built in 2014, quite some time ago. There have been a lot of technological leaps since then with plastic printing. The newest, well, not the newest, but the better alternative to plastic printing is metal printing. So when you print things out of metal, obviously they're much, much stronger than plastic. They are very, very temperature resistant in ways plastic can never be. Current tests, because it's a newly evolving technology, are proving very successful. There's a 3D printer that uses metal on the space station. It was built by the European Space Agency. and it uses a laser. And I'll describe how it works a little bit better with this. This is one of their first tests that they made. The way this works is a small robot arm extends a little thin filament of metal onto the part, onto this base plate here. That's what they're printing onto. And then a laser focuses onto that beam and melts it, and it kind of traces a path over and over. And it can print sideways and any way, because in zero gravity it doesn't matter because there's no weight. The newest form of printing, 3D printing, and this hasn't been tested on the ISS yet, but it has been tested in zero gravity like conditions, is cow printing, or computed axial lithography. It uses a small vial of photopolymer. Now, photopolymer is a liquid that when you shine UV light on it, it instantly hardens, or very quickly hardens, sort of like if you know what UV glue is. It's like that. They fill a small vial with this photopolymer, and they put the vial onto a spinning track or onto a spinning device of some sort. And then it has a beam of light shined out of it, out of a sort of UV projector. Here. Yeah. This illustration kind of displays it a little bit better. They have a projector show something onto the vial, and this spins, and this plays a video of an object spinning, and it sort of forms apart all at once, like a reverse MRI. It just kind of, each slice gets built all at once, and it makes a solid object in like 10 seconds. It's incredibly fast. And right now it's limited to pretty small parts, but it's still a work in progress, and it shows really, really promising results so far, especially because you can stick stuff into the photopolymer and you can print stuff around an object. So like that's a solid screwdriver handle or a hook like up there, something like that. This was something I didn't expect to be researching when I started the project, and later found out that it's actually become quite important with recent technology, is biologic printing. So biologic printing is where they take living tissues and they're actually able to propagate them in space. And the reason they're able to do this so much better in space is when you grow these small, they're usually trying to print organs of some sort. They're trying to regrow organs or tissues or something. Those structures are really, really fragile. And on Earth, they would just collapse and break. But in space, you can print them much bigger and much more complex without the risk of them breaking. And once they've matured, you can send them back to Earth. So human cartilage tissues have been successfully printed already, and they're currently working on cardioid tissues for like artificial replacement hearts, but they're alive, so they're not like quite artificial. This is a human knee meniscus, a whole one that was actually 3D printed on the ISS back to Earth. So it's possible we can have replacement knee cartilage within the next couple of years. And then lastly, this was something important to NASA's current mission goals, is placing a lunar base on the moon. All of these challenges that 3D printing in space faces are going to be similar to the ones on the moon. You know, you have extreme isolation. You know, you're not going to have all the tools necessary. You know, what if something breaks? What if there's an emergency? You need to have methods available to you that allow you to quickly deal with an emergency of some sort if one arises. I think that's about it.
Living cells alive, it will not.
The research I did on the biologic printing was pretty short. From what I could tell, they were designing this sort of, there was this big box and it had some sort of matrix they were growing it in, I believe. Either that or they had some sort of fluid that they could wash into it that was feeding the cells. I'm not entirely sure. There wasn't like a lot of great resources I had access to. A lot of it is behind like a paywall, or you have to have like certain access at like a prestigious university to access a lot of these research materials. So it didn't have like a whole lot to go off of. But I think that's how it was working. They were printing it in some sort of matrix, like a gel or something.
These are,
I might appreciate that. Yeah, to be cool to keep going.
Yeah. Design, just here like a back work up there. Good at 3D printer work that is designed to fake down.
Yeah, so the plastic ones, this one that they put up in 2014, this first one, this works both on Earth and in space. Because of the stickiness of the plastic and the way we have to actually get it to adhere to the bed on Earth, that's the same principle. As long as it's stuck to the bed, it actually prints exactly the same. And that's not true for the metal one because it has tensile stuff and you have to melt it in a certain way and things like that. But the plastic ones do work exactly the same, which was kind of surprising.
Why have they tried doing it in an anti-gravity setting on Earth?
For this one? Oh yeah, so there's a type of plane, they call it the vomit comet, that not officially. It flies way up almost to like not quite to low Earth orbit, but very, very high in the air, and then it plummets. And they get simulated artificial zero gravity for like I think like 20 seconds at a time, 30 seconds at a time.
it's,
It's not very long, but it's enough to where this thing can print something in like 10 seconds. So it's enough time to test it all the way through. It was really fascinating. And then think they're planning on sending up some sort of setup soon to test it on the International Space Station. Oh, wait. It's more fun to say.
20 seconds. And it doesn't.
Oh yeah, it will keep doing that. Yeah, I forgot to mention it'll plummet and then they go back up and then they plummet and then they go back up because they're not gonna fly up to do it once. They're just gonna keep doing it. That's why they call it the vomit comet. Yeah, well, because the speed too on the way down, that's weightless because it's but on the way up, that's 2 Gs. I think you had a question.
Filament drop or you.
I think for this one, do you mean this one? I know that for the plastic one, there's like absolutely no risk. It's just molten plastic. It's not, I mean, most plastic, I think for 3D printers, you have to heat it to like 215 C. Like it's almost not enough to burn you, you know? For the metal one, there was significant risk. This piece here took them actually, I think, a couple of months to print all this, and it's not very big, and that's because they were testing it a little bit at a time over and over because having a laser on board your space station, it's like if it can melt metal, it can punch a hole through your aluminum space capsule, so that's problematic. This whole setup was inside a box, inside a box, inside a box. It was very, very secure because this one was hazardous, very hazardous. This one I don't believe because it's in a sealed vial. You do have to figure out how to get the part out and clean it, which is, it's like it'd be kind of hard to clean it because you can't just like rinse it. I'm not entirely sure how they plan to do that yet. Oh, like in that? It's some sort of the one I was seeing is basically just like a video projector with like an LED. They didn't put a UV bulb in it, but that's essentially what it was. It was just a projector with that projected UV light specifically instead of visible light. From my understanding. Yeah, so anything that gets hardened in here. When you take that out, everything else in there can still be used. So you have to fill it up with the same amount of volume that left. And then, yeah, I don't think it gets like it's not permanently changed by like off casting or something like that. There is one that I've seen. Well, there's really two. There's like a main way, and then there's a different process that I saw. You can turn up there's a, when you print something out of plastic, it kind of creates a shell, and then inside that, it puts like a 3D matrix of like triangles or hexagons or whatever shape you pick, and that makes it so that it's not solid plastic. It's like a matrix inside. Depending on the density of the matrix inside and the shape that you pick, you can make it a lot stronger one way or the other. You can say, like, if you wanted to print a handle to something, right? You wouldn't print it this way. You just turn it on its side, and now the plane is that way, and that's the plane of force you're acting upon, and it makes it strong that way. The only other one I've seen, and this is an actual process that fixed it. I should have put a picture of it in here. Is a 5-axis 3D printer that can tilt its head like this, and it has a rotary base that turns around, and it can print something starting laying flat and then turns out like this to print on any plane it wants. And you obviously, the layer lines don't matter at that point.
You, think from the next project.
Yeah, I'm up again. You gotta give me a second. I grab my stuff. Lincoln was my partner in crime, was going to be here. He had prior engagements that he had to attend to. This second project was not sponsored by a program or anything. This was a personal project that me and Lincoln Roberts decided to study just out of interest. And it's on continuum robotics. Now, continuum robotics is defined primarily by the fact that you have a jointless, flexible structure making up the primary part of your robot. And when you have a flexible structure inside of your robot, that makes it so that it's very, very fluid. It's very articulate. And while it's less strong than typical robotics, it does provide more agility and more control over the range of motion. Right now, there's a lot of different applications for it. The major ones are something like robotics appendages. So like if you want an appendage on your robot to be very maneuverable. Medical applications. These are being heavily researched for surgery robots because of their precision and the fact that they can reach inside you. And for manufacturing purposes, if you want to do repair on something that's not really an easy shape to fix, like an airplane fuselage or something like that. This is the main difference between normal robotics and continuum robotics. With normal robotics, you have a different segments of this thing that are perfectly rigid and then joints in between, sort of like your arm. Your arm works that way. It's like we have rigid sections and then a joint. For a continuous robot or continuum robotics, you have a flexible piece all the way through the robot. And that flexible piece gets kind of tilted or turned using stress or using a pulley system, or you can use hydraulics, I believe. And this is another explanation, another way to see it, is you can have discrete robotics, which is, again, like your arm. You can have serpentine robotics, where you have many segments, right? And that will get you very close to what we're looking at today. But then continuum robotics is truly when it has full arc. There's no joint in it whatsoever. The whole thing bends and is actually flexible. This is a demonstration of what or how the actuation works. You have, or one way to do it, a lot of kind of vertebrae running up along a flexible spine, and then you put a cable through each of those. And when you pull on the cable, it bends it over. And then similarly, if you were to run a cable, now ours doesn't do this one, but it does do that. If you run a cable spiral wise and you were to pull on that one, it causes it to twist and flatten. And using those two types of movement, you can get to just about anywhere in space because you can bend over and then you can flatten and bend over in any sort of combination. And now we'll move on to our design. So we have our central flexible spine here. This is made out of a PVC pipe. And then we use these high tensile fishing lines for cable actuators. These vertebrae are 3D printed and they have holes running through them and they're fixed very tightly to the pipe. There's not any screws or anything holding them in place. They're just kind of friction fit. And then we have a couple of different motors, and each of these has a little winch on it. And the little winch either draws the cable in or pushes the cable out, depending on which way we want it to move. And then now we'll do demonstration. You are going to have to give me a minute. Oh, I should also say, this is to demonstrate the fact that you would have a tooltip of some sort on the end of this thing, like a welder or a scalpel or something like that. We didn't have time to make a thing on there, so just kind of imagine that this does something important. I'm gonna put this down for a second because I gotta calibrate it real quick before I turn it on and stuff. Every time I turn it off, the cables go slack, and then I have to tighten them up before I turn it back on. So those plug the motors in, and then not gonna want to turn it. So gotta make sure it's in this neutral position when I turn it on. Hard to turn the motors after that.
I think this at this point, at least for a good.
Some elevator music. Yeah. I think yep. Yeah, we're good. Something calibrated just a little bit more. Demonstration, or after all the presentations are done, I'm gonna have this over there in case anyone wants to take a closer look at how this works. Especially this gray box down here, which contains all of the actual workings of how it understands what data I'm going to send it from the computer, stuff like that. have a program here that controls these motors both at the same time and these motors both at the same time. So they spin the same way so that one unravels and one rolls up the same direction. Yeah. So I can jog this thing around and I can make it move in any different directions that I have a program here which and this program here is going to demonstrate its range of capable motion. And since ours doesn't have one running all the way up in a spiral motion, it can't flatten. And the material we picked doesn't allow it to flatten because I think it's too rigid, but it does demonstrate that it can fold over. If we wanted to have more complex motion where we had two curves, I would have another set of 4 motors that would run another 4 cables to another flexible section on the end of this. And that would allow me to get it to curve one way and then curve back the other way any way I desired. I think I have one more program in here that I can show you guys. It should wave back and forth, I think. And that's all. And then again, I'll have this over there if you wanna see like what's in the box, or I can show you. Oh, maybe I'll let people drive it around. We'll see. I'm gonna unplug this, get it off the stage for the next guy. Probably just building it was not hard, actually. I think the hardest part was coming up with and experimenting with all the different ways it was going to behave once it was plugged in and working. Because we only assembled it like almost the whole thing in the past like about month, and this has been like a year-long project. The hardest part was coming up with like, oh, if I pull this, how's this going to behave? It's kind of like a puppet almost. And I have to think about like, oh, is it going to curve this way? Is it going to curve that way? How big of a vertebrae piece should I use to get the correct torque or get it to twist? What if the motor's not strong enough? And all that work that me and Lincoln did paid off because this was actually like the first prototype. We tested this out for like the first time a few days ago, and it all behaved exactly like we had designed because of all that preparation and thought that we'd put into it. Yeah, Gabbard. No. The vertebrae is, like I said, it's 3D printed. I made those myself. A roll of filament costs like $15. This PVC, this is flexible PVC pipe. We found this online for I think another $15. This set of 3 motors, I had this motor laying around, and then we needed a few more, and I believe those cost 20 or so. $30 for the motors. I found this wood laying in Tioga 4, so I took it. And then the wires I just found laying around. And my dad contributed this wonderful box here with in it. Um, it does have a name. The name is related to cigarettes because we were joking that it's meant to light your cigarettes. So we felt that was in poor taste. So we're just gonna call him Chompy. Oh, yeah. I'm not sure. This was more like a tech demonstration. We wanted to see what challenges or like how the technology worked. I think for this, it's not precise enough or strong enough to have a realistic application, I would say, beyond just like an educational demonstration, which is fine. That's kind of the end goal for this, I would say. But if I were to build one, say, like out of machined metal and I had high torque motors or probably hydraulics, this could take on some sort of, a manufacturing task, I would say, or one that's mounted to the ground.
As not that, as this is going on, my brain is spinning. So, we know, we have some going to other institutions that we go woman with. Next year, the potential to continue this on, you guys, or having ideas as to how you might continue on for.
That's a great question. I know I'm transferring to Oregon Tech in the fall. I'm probably not going to pack this with me. I'm probably going to make space in my car for something more important. Uh, but if Lincoln would like to, or someone else would like to keep working on this and actually get a working tool head on this, because it has parts down there for it, and the fact that we used a pipe means I can run wires up that. We even thought about having pneumatic stuff. Since we already have an airtight pipe, you could fit a pneumatic fitting into the bottom and have, for those of you who have ever seen like pneumatic clampers or something like that, or a suction cup, those are pretty common in manufacturing of more delicate objects. You could do something like that if someone wants to continue working on this, because it works pretty well. I would say surprisingly well for what it is. No, so this has an Arduino in it, and it's running a program called Gerbil. And then I have Universal G-code sender on here. So all I have to do is, it's really easy to use. I just type in commands, and then Universal G-code sender. This is like meant for like 3D printers, laser cutters, that sort of thing. It's not super designed for this, but I can send it a command. The command goes to the Arduino. The Arduino interpolates the command, sends it to a motor driver, and the driver powers each motor and gives it a pulse. So like for the circular thing, literally to make that program, I opened Notepad and I typed in a list of numbers. I said 8 0 0 0 8 negative 0 0 0, and it just does those.
Or is it?
Yeah, at the start of the program, you have to specify, there's a command to tell it, oh, we're in millimeters, and I'm using absolute references, because otherwise it'll say like, oh, go this amount, when I want it to, I want to tell it to like, oh, 8 within reference to 0, I want it to, and it thinks, it has a picture up here of demonstrating what it's supposed to be doing when I run that program, and it's way off because it's basically drawing a square when that's not really what it's doing, of course, because it's rigged up to wires or pulling it around. But it's just kind of, it's meant to go on like a linear motor setup, something like that. If I wanted to, and if I had more time, I could probably tailor it to where I could have like an inverse kinematic setup where I could tell it, oh, go here in coordinate space, and like have a program to tell it like, oh, that means I have to go by this much over to here. And that's what you would do if you were going to use this for a realistic application. Because I'm not just going to guess. I'm going to say, oh, my part is here. I needed to go here. And then it'll do all the work to find out what it needs to do to get there with a limit switch or something. I saw a lot of people using Hall sensors on more professional setups to determine like, oh, how much is it bending? Because it's hard to measure like how much something is curved.
James, information on Tuesday title data.
You won't hear me? Do I need the microphone for this? No? Okay, can you guys hear me now? Hello, my name is Mark James. An electrical engineering student here at SWOCC. And I'm presenting our collaborative research project with the University of Oregon, the Lundy Tide Gauge. As you may know, we live in Coos Bay, Oregon, and we are directly above the Juan de Fuca subduction zone, right where it meets the North American plate. Under our feet right now, we are experiencing massive geological movement. As you can see, we're somewhere below this, but the Juan de Fuca runs all the way down to California. You might be wondering, how does this relate to us? Well, we have recorded tide data from the north in Newport and south in Brookings, and they both show a reduction in sea level relative to our NOAA tide gate in Charleston. So our team from U of O and from here at SWOCC, we had a few questions. We were wondering, what if the ground beneath our feet was subducting at the same rate as our water level? And would that justify why our tide level is not shrinking as fast as those to our north and south? And what if we could measure water level at the same time as measuring geological movement? So Professor Dean Lively Brooks of U of O and Ray Weldon of U of O had an idea, and they said, what if we built a tide gauge that was tied to geological survey points? Then we could measure tide data with direct correlation to ground movement. And Ray has found historic tide data that dates back to late 1700s. So if we could make a timeline from the late 1700s to our modern time, we could basically map our tide differences from then to now. That brings me to the staff. Yep. So our first prototype is the staff. It's pretty rudimentary. We have a very sophisticated measuring system that me and Nick Weldon have installed out on the Lundy docks, and we have a camera which is located in this box, and it's taking pictures of that very sophisticated staff as the tide is raising and lowering. We can then take those pictures from that staff, run it through a MATLAB code that Professor Lively Brooks has made, and we can convert that into numerical data, and that has given us some fluctuation in our tide data. This is just a segment of the data. It's been running since late January, and it's been going pretty steady since then. So we have the camera back here in the box, and alongside, oh man, I got pictures all over the place here. So this is on the Empire boat ramp, and over here towards land, towards where all the concrete is laid, we have geological survey points, and so those are going to be measured by the state, and we have references to exactly how far those are in coordination to sea level, and that's how we can tell you the tides, and that's how we can tell you we're at an 8-foot tide or we're at a negative 3-foot tide. So our camera system, it takes pictures every 6 minutes, and we run that through the code, and it gives us our tide data. This is our calibration data from that tide. So this is basically what's telling us how far our pictures are from the top of the staff. And we've had quite a few problems in doing that. We've ran into algae buildup on the staff, driftwood. Someone has to manually take all of that code and put it through a program and then take that data and then graph that data. And it's a long process that Nick has been working on, trying to solve all of those little problems. So we came up with a new idea, and this this shows us where we see these anomalies in our tide data. Like right here was a log drifting in front of the staff, so you could not see the staff day or night. Our camera is infrared, so it does record at night, and it has a month-and-a-half battery life, so it's relatively low maintenance. But someone has to scrub the post, and someone has to take the card out, and someone has to code the data, and someone has to turn it into this beautiful graph. So we came up with a new idea, and it's inspired by some Aussies down under. And we have an Arduino tide gauge that runs off a ultrasonic laser sensor. And this one that you see in the picture is battery powered, and it only has about 5 hours of battery life, and it can only measure about, I think this one's 4 meters. So our dock is too tall for that, and we don't have the time to have someone go out there every 5 hours and change the batteries. So our newest system that we're working on right now is a, oh man, I wrote it down. It's a solar-powered ultrasonic sensor system, and it runs on 5 volts with a stronger sensor system that can measure 24 feet, and it's 2 millimeters accurate past 30 centimeters. So we should have no problem with battery life. We should have no problem with algae buildup or driftwood because it's all enclosed in the staff, and we won't need to worry about running it through the code and having someone pull the SD card out and program it and then running it all through that just to get our data. So yeah, it's currently in development. I've got schematics going right now for it. And that's about it. Yeah, pretty short. Any questions? Yeah.
How powerful they're doing.
Yeah.
As and whether it's not necessarily.
Right. Yeah. So yeah, so it runs from solar and then it will run into batteries that have been donated to us by Mr. Pruitt. And yeah, so those should last us, I mean, at least 2 weeks with the battery life that those batteries have. And then if it doesn't, we can always put more batteries in it. Yep. Yeah, let's check it out. Which one? This is, yeah, so this is just measuring the distance from the top of the staff to the camera. So each one of this is measuring the distance from the camera to the reference points on the staff. So this is basically so like the red X is going to be the number on the staff, which is a prototype. And then the blue is the accurate measurement of the distance of the tide. So we have some skews there, as you can see. So that's part of our problem that we need calibration. Yeah. You know, I didn't make this graph. I couldn't tell you. So we're hoping that it can measure our tide data and we'll be able to survey it to these geological reference points. So our goal is to measure correlation. So as the tide is raising and lowering, we'll be able to tell if our ground level is moving with that at the same rate. Because NOAA, it's the national, yeah, yep, you got it. Yep. They have their own tide gauges that measure all these different things. They measure tide and they measure wind and temperature and salinity, but they don't measure ground movement. So the ground that those gates sit on could be raising or lowering, and we're in the subduction zone, so they're probably lowering, but they don't account for that kind of measurement. Yeah, so when we look to the north in Newport, and we have our data in the south from Brookings, their sea level, like that they can see, so every time the tide raises and lowers, that neutral zone that it comes back to is lower than our neutral zone where ours comes back to. And so our hypothesis is that our ground is sinking. And so that's why we're not seeing a difference in that fluctuation. They are, and they're farther north on the plate and farther south on the plate than we are. Please. Thank you.
Is your ground level drops suddenly a whole lot.
Right.
And then the tsunami is way worse than it would have been
Right.
otherwise. But if you are actively sinking a little bit, what are called slow slip events, likely won't have a catastrophic ground drop and a Cascadia. And therefore, it's bad, but not as bad as for people in Broken.
Right, which is concerning thing to see in like physical data from the government, that our data is not showing the same across the whole plate. So as that subducts, we are getting more and more friction, and to see a drop in water level is significant enough to want to find out why, especially with, yeah. Yeah. Right. Right. Correct. Exactly. Yes. Anybody else? Cool. Awesome.
Since we did have one more slot for Professor Watson's couple of geology pieces, they were challenged if they wanted to get present here. But to me, really would like to ask. I will give you a couple minutes to talk about what they were working on. You want? It's really because it's not a could not hear that. When my high school turned Trella did over fall and part of winter quarter, is she basically looked at hazard preparedness for geologic events for people. And for the investment. What was out there, and how do these different groups communicate with each other? And a lot of what she found was that while we do have a lot of different organizations that are working with monitoring possible hazards, ranging from landslides to Cascadia megatrust earthquakes to tsunamis, things like that, the communication between groups, the public is very, very poor. So what she did was she made a little pool that she got a whole bunch of high schools to take home to their parents about, do not sign this day. And 5% of the polled individuals from Goose Bay have no idea that the decommissioned and that we no longer have tsunami this now. So definitely it sort of raises some awareness. She also prepared some educational materials, and one of the things back on post-grad graduation, one of the display cases down here will be switched out to be Mount St. Helens and Cascadia 1700 earthquakes as examples of like full disasters that have
Pacific Northwest. And so Trello will have some, she made some very beautifully artistic, but also little, like conservation signs. So those will be hanging up there. And the other, my class does I challenge them rather than writing a paper, communicate via a variety of creative projects, something about a geological time period. So I how we're working on their projects, so because it's not due till final stage. But my historical geology students take a time period into the major events that happen geologically, how climates change, and what kind of settings. So there probably cool art that will also make it into various displays from students. Because I get hate like do like podcasts and some students are curious. They do cool work. So all the students come back from as we further they can ask their respective students there about the projects. I know Sabrina does have her poster in the back to look at as well. Mason will go through the robot and feel free to ask whatever questions you may have had, but I was going to ask during the live stream portion. We thank everybody for participating. And this is one of our favorite events to do every year because we get to showcase what our students have done over the course of the year. And here with me, let me take your picture real quick. Did research. Thanks. Have your favorite time question. The sign in on the sign-in sheet. We go to Filipino. We are currently actively working on the that is a ongoing project. Of course, there's stuff on our STEAM Academy as well as our SSC program fundraising goal to send SWOCC clients to the handouts on that. Get if you got a copy of college, actually stuff also on the back of the so you can there as well. Well, and our question series kind of takes the summer off, but we'll have additional lectures in September to college the various college websites. If you have a Facebook page for more information on all of those. That feel free to contribute.